Synthesis of silver nanoparticles for the spectrophotometric detection of vitamin B1. Abstract: vitamin B1, a nutrient belonging to the B vitamin complex, is indispensable in minute quantities for the proper functioning and sustenance of the body, aiding in enzymatic activities, sugar breakdown in food, and promoting nerve and heart health. According to recent reports, this vitamin, along with various other drugs and supplements, can accumulate in the environment, posing a threat to both human and animal health. In fact, excessive vitamin B1 in the body can expedite the growth of cancer cells, thereby necessitating the development of new detection and quantification techniques for these compounds. In this regard, noble metal nanoparticles have emerged as promising candidates for the colorimetric detection of numerous analytes. Owing to the collective oscillations of free electrons in response to incident light, also known as localized surface plasmon resonance, LSPR. The LSPR spectrum of metal nanoparticles varies with their chemical environment. In this study, we present the synthesis of silver nanoparticles through chemical reduction and their application in the spectrophotometric detection of vitamin B1. The results demonstrate a linear response of the absorbance of the LSPR of the silver nanoparticles with the concentration of vitamin B1 in a range of 20 to 160 micromole per liter. Methods for the synthesis of nanoparticles: 48 milliliters of an aqueous solution of sodium borohydride at a 2.00 x 10 to 3 molar concentration and sodium citrate at a 3.55 x 10 to 3 molar concentration were prepared with vigorous stirring for 30 minutes at 60 degrees Celsius. Then 2 milliliters of silver nitrate were added drop by drop at a concentration of 1 x 10 to 3 molar. Then the temperature was raised to 90 degrees Celsius. The pH of the solution was adjusted to 10.5 using sodium hydroxide at a concentration of 0.1 molar and continued heating for 20 minutes. The preparation of a standard solution of vitamin B1 was continued. For this, the vitamin B1 was weighed, dissolved in 40 milliliters of distilled water. It was carried to ultrasound. Later it was filtered and the solution was taken to a 100 gauge flask. Milliliters and fill up to the mark. Table 1 of samples shows the different concentrations of each sample. Results: Figure 1 shows the UV-vis spectra of the nanoparticles synthesized and placed in the presence of vitamin B1. The absorbance of the particles shows a gradual decrease as the drug is added, indicating that a calibration curve can be drawn. Figure 2 shows the linearity of the vitamin B1 concentration against the absorbance response at 400 nanometers, which fits a straight line with a coefficient R2 equals 0.9468. Figure 3 shows the silver nanoparticles characterized by means of TEM. In this case, particles with spherical morphology and an average size of 27 nanometers were obtained. Figure 4 shows a possible interaction mechanism of silver nanoparticles with vitamin B1. The presence of vitamin B1 can cause an agglomeration of the silver particles, due to the nitrogenous groups of the drug molecule and its high affinity for silver, which leads to a change in absorbance. Conclusion: the silver nanoparticles were successfully synthesized by chemical reduction and used for the spectrophotometric detection of vitamin B1. It was possible to synthesize particles with an average size of 27 nanometers, this according to the TEM image. The results demonstrate a linear response of the absorbance of the LSPR of the silver nanoparticles with the concentration of vitamin B1 in a range of 20 to 160 micromole per liter.